So what I'm trying to do today is actually talk first a little bit about rabies virus, because that's, the old love since 25 years, and then go from rabies virus to Ebola virus, which we work for about 5-6 years now. So rabies virus is another neglected infectious disease. It's a well-known disease actually to mankind, but it's a huge problem worldwide. And what is probably not well known. So this is actually also a very simple virus. It's also a negative stranded RNA virus, which are all very similar organized. They have a host cell. They have a host cell-derived membrane, so they all envelop viruses. They have a negative stranded RNA as a genome, which is only infectious and an infectious template when they encapsulated in the nucleoprotein. This protein, they have a phosphoprotein, all disruptoviruses, which actually is a polymerase subunit and also interferon antagonist, a matrix protein, and a single glycoprotein here. And the beauty of these viruses is actually that you can actually replace functional this glycoprotein here, or you can add additional glycoprotein. That's basically the vaccine approach. But before I actually come, so all of you know this here, classical rabies virus, genotype 1 is responsible probably for 99% of the human deaths, but what you probably don't know is there are 16 other genotypes which go in 2 filler groups, and a lot of them are actually also emergent infectious disease. And in red, that's all the viruses we don't have a vaccine against. And your attention, they also in Africa, and especially Mucola virus in South Africa is a problem. And there was human death. There's no therapy and there's no vaccine. But all these viruses are very similar. Basically, you can't distinguish you by disease. If it's a classical rabies virus or Mucola virus, very similar disease. Just antigenically, they're different. And most rabies viruses are highly neurotropic in the infected host and cause a fatal encephalomyelitis in mammals, and that's without vaccine. And actually, rabies is, after infection, probably the most lethal virus, which kills 100% of infected people and animals. So hosts are all mammals. In the developing countries, 40 to 70,000 die from the disease each year. Probably the numbers are much higher. This is based that in Africa, we really don't have a good reporting system. And when they checked in Tanzania for so-called malaria-induced encephalitis in children, 10% was actually rabies. That tells you about how good that system is. It's rare in the US to post-exposure prophylaxis. A good thing about rabies virus is that you normally know when you got exposed, right? You get bitten by an animal. The bad news is that the bat-derived rabies in the US, a lot of people actually do not realize when they get exposed. That's why they tell you if you find a bat within your bedroom, catch it and let it analyze or get rabies vaccinated. The lucky part about the US is, again, that all bat-derived rabies virus in the US is classical rabies virus, so the vaccine actually will protect you. So that's just to walk you a little bit through the numbers. So there are different, the US is very special with rabies. So we have different species. We have raccoons, skunks, we have foxes, and we have bats which transmit rabies virus. And here's the numbers. I'm kind of the emergency area for rabies. I mean, it's unbelievable how much rabies we have. Basically, if you find a raccoon or a skunk in your backyard, there's a good chance that they have rabies. The numbers are really high, and I have to say the USDA really failed to do anything vaccination-wise, because Europe got completely rid of rabies by vaccination, where I have to say foxes are very easy to vaccinate with live baits, so with live virus. It's a little bit harder for skunks and raccoons. This here shows you now the skunk numbers here, and more interesting is actually, this is a little bit when you look you will find, but that's bad rabies virus, and that's the cases where you have to be careful because you really don't know if you will be exposed or not. And we want to study a little bit bats. So in bats, rabies is not as pathogenic than in other mammals, which it couldn't because they live so close together. If one has rabies, the whole thing would have rabies. So that's interesting. But again, worldwide distribution. Here you see rabies is actually everywhere, except here it got actually eradicated by vaccination. So the highest numbers are from India, but that's only because they have a relatively good reporting system. Africa, we really don't know. Rabies virus infection is relatively simple. You have normally transmission by bite. You have no host response to the infection. Normally it goes, we don't know where it replicates first. It's a non-lytic virus because it has to go up a motor neuron. We know exactly how fast. It's about 1 millimeter each hour. It travels up, so you can calculate from that when it will reach your brain. It moves to the CNS and then it induces behavioral changes. I mean, it's all pretty fascinating for 5 genes, you know. It infects actually areas which normally are in charge of your emotion, and that makes aggressive behavior. And then you have the 2 forms. This is an older picture. So if you have symptoms of rabies, there's really not a lot we can do for you. They just re-stained people, and so now we put them in a coma, and you basically wait until they die. There's some publication about therapeutics, which I think are not really working. And that shows these 2 phases, the furious ray and the dumb, where it's more lethargic. That's more like it is in humans. You really don't have the furious disease in humans. There was a case of a lion with rabies, and they get the furious disease. That's really not what you want, a rabies-infected lion, I guess. So basically, pathogenicity of rabies very, very shortly. Pathogenic viruses replicate really slow, spread fast. They are less immunogenic. Basically, they don't make a lot of protein. They're just sneaky. They stay under the radar screen of your immune system. They go into the neuron and go up to your brain. Then we have 2 things which are also important: neurotropism. Pathogenic rabies viruses have a high specificity to infect neuronal cells and no other cells, which makes sense. And then this neuroinvasiveness. This is the ability of a virus to enter the CNS. The strange thing is all these bad viruses are not really neurotropic, but they're highly neuroinvasive, and we have no clue how that works. So there's still a lot of research to do. Now we come closer to the vaccine. So rabies virus is actually a good vaccine. The story is that I put it in your stomach 20 times. It's all not true. In the meantime, it's IM, like your flu vaccine. You get 3 inoculation at days 0, 7, and 21 to 28. Then you're normally immune for the rest of your life. Some people have to be boosted, some don't. Because we work with rabies, we get blood drawn every half year and tested for neutralizing antibodies, which predict if you're protected or not. Good thing about rabies, you even have standard, you have international units which predict if you protected. And then about 30 to 50 million of the vaccine got used, which is an argument for our combined vaccine. Post-exposure treatment, you know, rabies is one of the diseases where you actually can do something after the fact. What you do is active immunization and passive immunization at once. So it's a human IgG sera. So it's really from volunteer which they vaccinate and boost several times, and that's human sera. There are also some neutralizing antibody which may replace that. It's very expensive. It's $2,500 is one treatment with the serum. And you have 30,000 post-exposure treatments in the US alone, so it's good business, I guess. Incubation time, 3 to 8 weeks. In general, there are description of cases after 2 years. It's true, but in general, mostly it's 3 to 8 weeks. The incubation time, and after that, mortality is 100%. Again, furious and done. which means you have this hyperexcitability, that's aggression, that's how the virus actually gets transferred by the aggression and biting. In both cases, you have that by cardiac or respiratory arrest. And what is really interesting on rabies 2, and I just briefly want to show that before I go actually to our vaccine approach, you really do see not anything. And there was, I don't know if you heard about it, cases where they transmitted rabies to 3 or 4 people because they thought somebody died of a stroke and they transplanted the organ and that person actually died of rabies. And the reason I have to defend the doctors here a little bit, you don't see any pathogenic changes in the brain. So if you look, that looks like the normal here, you bicuspid, you have it full of rabies antigen here early on, but you don't really see any apoptosis or damage of the neuron. So that's really a hallmark of rabies virus. And what little bit research on that, how does that happen? How does the virus get cleared from the brain, or how does it affect the neurons? And there was one theory, a neural cell that hypothesis or neural dysfunction. And to analyze that, we actually used that Cree reporter mouse system, which has this gene, so every cell is red. And if you cut that out by expression of the Cree recombinase, they turn to green, from red to green. So basically, if you infect with a rabies virus expressing that protein, you change to green. This is a rabies virus with a Cree recombinase. This is some reporter cells. If you infect them with regular rabies virus, they stay red. And if you infect them with a Cree, they change to green. The problem is really if a cell gets cleared after the infection, you won't find it anymore, right? So you can't look for the antigen. So if you use a not so pathogenic rabies virus and you look in a mouse 2 months later and it survives, you really don't know what happened to the cell. So with this system, we want to solve that problem, and it actually worked better than I thought. So that's the infected brain over time for mice surviving. So we used the vaccine strain of rabies virus. At day 7, day 15, you see this infection. So that's all infected cells which change to green. After 1.5, 3 months, after 6 months, you see all the cells are still there. And we actually sorted for green and red cells and showed that this neuron had functional changes. So what that means is actually that we want to look a little bit more into that, because that also tells you about something about therapy. The damage may be already done in this neuron. You probably can't just restore the function. But now I want to go back to the vaccine against Ebola virus, starting with this rabies virus, a worldwide risk. But these are all areas where you actually should get vaccinated if you go in this area. I mean, not in the cities, but in remote areas. And that's where acquired rabies is a high risk. And that was our starting point before we actually had the last Ebola outbreak, because there was vaccines out there, but financially none of them were really attractive. So we thought combining basically 2 vaccines in one would be a good way to address a problem. Filoviruses, I really do not have to go in detail through that because Erika really did a good job to explain all of that. We have basically 3 main candidates: Ebola virus, Sudan virus, and Marburg virus. They now listed as separate viruses before they were together. And as I said, also enveloped viruses with a genome, 7 genes, about 15 kb. This shows you such an Ebola virus. A fascinating thing about Ebola virus, that it's a really long virus, about 4 or 5 times longer than rabies virus, with this filo-like structure. And when we talk about vaccine, I think the field in the meantime agreed that really this glycoprotein, which is on the surface, is the best vaccine target. I think that's at least one where the field can agree on. If we look a little bit into the epidemiology, I mean, it's not really new filovirus outbreak. It was not predictable that it will such a big one, but I mean, 1976 we had the first outbreak, and then we had outbreak every year. A little bit it was predictable that at some point it will explode, because most of this previous outbreak were really in remote areas which you could contain very well. So the World Health Organization, CDC, went in and kind of made sure that everybody stays in these areas. So today, with people more traveling, and it's actually well established that it actually probably started with one single 2-year-old boy who got it probably from bats, and then it spread through funeral in a large area. That shows you the area, mostly Guinea, Sierra Leone, Liberia, we're certainly at the borders. I mean, every country really, really wants to be not having any Ebola cases, but the borders are pretty often, so I think there's certainly some spread outside. These are the newest numbers, so the number seems not to be rising as bad as initially was expected, but it's still pretty high. That's updated here that shows you I can't spell January. So 20,000 cases, about 8,000 there, and 13,000 confirmed cases. This is really important, and I don't know if people appreciate it, that the confirmation of these cases is so important, because West Africa has a lot of hemorrhagic diseases. And if you would live there and have Lassa fever, which is not as deadly as Ebola, or you have yellow fever, or you have another hemorrhagic disease, you have malaria with high fever, you really don't want to get together with the people having Ebola. So the diagnostic system is really, really important, that these people trust to go to the hospital for treatment. Okay, and that's the case numbers. Briefly about the pathogenicity, I think it's well established what happens with Ebola virus, that you have entry into macrophages and that it uses the lymphatic system to spread all over your body. It replicates very well in lungs, kidney, liver, and at some point you just have this really, really high viral load, and you have this organ failure from the kidney and the liver, and that results in that. It also, the endothelial cells start to get leaky, but it's not always a hemorrhagic disease per se. I think only 50%. It doesn't matter, it's mostly organ failure, which results to death. It's not airborne. Here's the lungs of people. It's really not an airborne disease. You really have to get in contact with body fluids. But at the end, where people die, the viral load are really extremely high. You have 10 to the 7 viral particle. I mean, this is a lot. So if you touch without protection, you have a good chance to get infected. But it's not airborne. So this I really can skip, because Erika already told you all about the receptor and the macropinocytosis, which I think is interesting by itself, because that's not how viruses normally affect. And that you need this cleavage shown here, and this is actually it's the NPC1, that's the only identified receptor, which is essential. This other one here, initially, this host factor binding, that already tells you host factor binding is not very specific, are all replaceable. So that's really the important binding. What Erika didn't mention is there's also something else about Ebola, not for Marburg, but Ebola, which tells you the importance of the antibody, because the virus takes a lot of effort to make soluble glycoprotein, which may be misfolded and which is also maybe a target for antibody, just to deplete antibody. So basically 80% is that short soluble protein here, which is just produced and pumped out, and only 20% is a real protein. And we know if you change that and you just have GP full length, which happened actually in some of the vaccine study, you have a highly attenuated virus already. So the soluble protein is important. So in general, that together, the data which I presented was making the field thinking that antibody against GP is probably how you can protect against Ebola virus. So I run you a little bit through the Ebola vaccines. And I want to point out again, antibodies, you have virus-neutralizing antibodies, but you also have antibody-dependent cellular cytotoxicity, which I think is actually a major mechanism of protection. You can have this antibody obstination. Do you need antibodies? Do you need B cells? So are circulating antibodies enough, or do you produce antibody? And then certainly the biggest controversy is the function of CD8 T cells. Do you need CD8 T cells, or do you need just for clearance, so basically naive one, which amplify and just clean up, or do you need memory T cells? And it seems to be a little bit different for different vaccines and how well they work. So front runner, we have two front runner. You all heard it. So there is a chimpanzee adenovirus vaccine, GSK vaccine, which is basically mostly that work was initially all done by Gary Nabel, and actually Nancy was the driving force behind it. That shows you one that it's a replication deficient virus. And the concern was, as you know, from the HIV, adeno5, a lot of people have antibodies, so the vaccine really doesn't work well. And if you put it in certain people, at least for HIV, it can even increase infections. So not a good vaccine for Africa, probably, where HIV is prominent. So people started, and actually that was initially done for the HIV vaccine, to this chimpanzee adenovirus, and that chimpanzee adenovirus 3, and there's a 63, and shortly that was shown. If you use that adeno 3, that's the best one, you give 10 to the 11, that's what I actually used in the human trial too. Then you get a good response. 10 to the 10, you don't get such good response. The problem, and that was recently actually published on this vaccine, is that the antibodies go down over time. So after 6 to 8 months, you lose basically your protection, and you have to basically boost, and you can actually boost with the same virus you need here, the MVA. So that would be the approach. I still have to say for emergency vaccine, that probably would be okay. 10 to the 11, but for long-term vaccine, GSK has to work a little bit on that, and introducing another viral vector won't be that easy if you have to prime boost with two different ones. So the next one, which you heard a lot about, is probably the vesicular stomatitis virus. VSV and rabies basically looks the same. VSV is also a little bit nootropic. And you see here this extremely busy table. That just is here to show it was extensively tested with titers to 10 to the 7, and they always had protection. They tested it against Sudan, they tested it, and it is a very good vaccine. But it is a replication competent vector. So as I told you before, for rabies virus, you can functional replace a glycoprotein. So that's vesicular stomatitis virus. You take out its own glycoprotein and put in the Ebola glycoproteins. And you have a replication VSV, which uses the Ebola GP to infect cells, which works well, but had some, recently it was tested that the incidence of joint inflammation was very high. So in the second part of the clinical trial now, they go back from 5 times 10 to the 6 to 300,000, so 3 times 10 to the 4. So that's certainly a 100-fold difference to that. So we have to see how the antibody responses are, but we don't have any information on that yet. And here are the other approaches, because in the press you hear only about the two. There's also, initially inactivated virus, probably not a good idea. and it really doesn't work well either. But there are certainly virus-like particles which you make from this protein which showed protection, DNA and recombinant adenovirus. Vaccinia virus actually didn't work at all, but there's a paramyxovirus, human cold viruses mostly, which are used and work well. There certainly is a concern about pre-existing immunity. VSV works well, and there the rabies virus, which I will spend some time now to talk about our vaccine approach. So in general, for vaccinology, the general slide is that's our problem here. So you have a replication-competent vectors. They normally really immunogenic, right? Replication deficient, they less. So because they can spread within the host and they keep on spreading until they cleared, so they're normally good vaccines. Mumps, measles, rubella is example for replication-competent viruses which work well. Replication deficient vectors, I don't think we have anything as current vaccines, but we certainly have deactivated viruses like for the rabies virus, or chemically killed virus. We have virus like particles, certainly, VLP. Hepatitis B is a VLP produced in yeast, works actually pretty good. Mostly you need 2 inoculation. Then we have DNA vaccine, and then we have protein itself, which is mostly not really immunogenic without any adjuvant, because there's no really good reason for your body to make a response against a single protein. And then to get these things better, all this here, more immunogenic, then you add adjuvant, and you have at adjuvant you go, it's a risk area again. So it's all a balance and try and error to make a good vaccine. So our approach for rabies virus, the thought was, and as I said, it was 4 or 5 years ago already, to generate a recombinant rabies virus-based vaccine vector that express a glycoprotein of Ebola virus. We thought that a desired vaccine, Ebola or Marburg, with approved financially viable vaccine such as rabies vaccine would be a good idea. Justification, at least 24,000 rabies deaths in Africa. I mean, if you think about that, that's yearly numbers. That's still way higher than the Ebola. And the other idea was live attenuated rabies vector vaccine could be dispersed to Africa to put the primates. You know that primates are major victims of Ebola outbreaks. The numbers are even higher. One outbreak killed about 5,000 of them. So oral vaccine for something like that is certainly something we would like to consider too. And we saw that pre-existing vector immunity safety and high costs which have hindered previously a vaccine would be avoided by this approach. So that's our vaccine candidate. Rabies virus, as I said, 5 genes, NPM, and here's a glycoprotein. We further attenuated, so this is already the vaccine vector used for all immunization. In Europe, we put an additional mutation in the glycoprotein, which prevents the infection of neurons, at least the preferential. And then we just popped in the Ebola glycoprotein into this vector. We also made one without G, similar to VSV. Ours didn't spread for whatever reason. I have not a good explanation for that. And then this approach would be taking these virions, inactivated by BPL, which is a normal method to deactivate the current rabies vaccine, and use the killed particle as a vaccine similar to rabies virus. This shows you some EM studies of our particles. We labeled with 2 different gold particles. The larger one, this would be the vector, are directed against this actually is it's a larger one against the vector, and the smaller one here against GP. So you get labeling of only glycoprotein, and here you get labeling of rabies glycoprotein and Ebola GP. So you see this protein is incorporated. This is a little movie showing the same thing, just enhanced by color. 2 of this doesn't want to play. Well, sometimes it doesn't work, sorry. Anyhow, you see here the viral particles in green, and you see here's the rabies virus glycoprotein, in red the Ebola GP. So this virus we constructed. I just show some pathogenicity data before I show the monkey data. So what you normally do if you have a virus and you want to eventually use it also as a live virus, you do different inoculation for the potential of this virus to invade the CNS and for spread and neurovirulence by direct inoculation into the brain. And basically what can you see here is that after IP inoculation, the weight of all the mice goes in the right direction, so there's no pathogenicity. Same after IM. And here it's interesting, this is now intracerebral. I told you before, our vector without this 333 mutation is still neurotropic, even it's a current wildlife vaccine. And so you see, if you put inject it direct in the brain, the mice will die, lose weight and die. But if you add this 333 mutation, you have a perfectly safe vector which doesn't spread within the brain. Now we go to our first non-human primate experiment against the vectors. And if you memorize the color, it will be easier. So BNSP333 in black is always a vector, the empty vector. In red is a live replication competent. In blue is a replication deficient, because I told you our GP virus doesn't spread, infect, but doesn't spread. And in green is basically the red one, which was inactivated by BPL, this one. We had 4 groups of monkeys. 3 control monkeys, which got the vector 5 times 10 to the 7. Group 2, 5 times 10 to the 7, so replication deficient. 10 to the 7, that's the highest titer we got, on 250 micrograms, 2 inoculation. This replication competent, you can do only 1 inoculation, because after the 1 you have rabies immunogenicity, and then you can't really do a second inoculation, which is also an advantage of this, because you can use that multiple times. Here you see prime, boost, and then we challenged. Because we used rhesus macaques, we challenged 2 controls a little bit earlier just to make sure that our challenge virus really kills these monkeys. And here is the other control and the vaccinees. And then we follow basically by standard the animals with blood draw and analyze the immune responses. And that comes here, and this actually controls here. Sometimes monkeys, like humans, it's rare, but they survive Ebola infection. So that was from Heinz Feldman, a controlled sera for our ELISA assays, which I show you here at day 0, 7, 14, 28, 35, 42. And that's the groups of the monkey. So this you would think if you reach that level, you should be fine, because it was a surviving animal, at least if antibodies are the major effector cells here. And you see at day 0, there's nothing. Then we immunize at day 7, you see a little bit, goes up at day 14, day 28. And this is with this antigen, the immune responses just keep on going up over time. Because the antigens are pretty stable, this viral particle. Here at day 28, we boosted this green group, which was a replication deficient. You see it reaches, that's what made me happy, at least for some days. I'll tell you the end of the story soon. So, but that we would think that's about the similar antibody responses we need for protection. As day 42, that's when they were transferred for challenged. So now the big question is, vaccines is you immunize and then you challenge, and the readout is survival, right? And that's what we did. And here you see post-challenge day. So they were all bled at the day of the challenge. And here I give away the results. Survivor is a red box. And what you can tell here also, vector immunized animals didn't survive as expected. All the live viral vector animals survived. Here you see this. 2 survived of the replication deficient, and of the killed, only 2 survived and 2 died. So if you follow them longer, you see day 16 after challenge to day 28, you really get nice immune responses. You challenge and the immune response goes up and up and up, and the protected antibodies, animals end up with a lot of antibodies. So that seems to be really the measurement of protection. The problem was, and while, before I showed it I just recently found that paper, which I think is interesting because it lines really nicely up the viral load. So if you do a non-human primate experiment, you know if your monkeys reach a titer around 10 to the 5th, 10 to the 7th, you probably won't save them. It's more humane actually to sacrifice them at this level. And if you look here, it's really the viral load is clearly an indicator of pathogenicity and survival. So this is actually, these viral loads are from human, and you see survival rate from about 30% if you never detect a titer higher than 10 to the 5th, and really high death rate if the virus goes higher, and that's pretty much what you see in non-human primates too. This here is completely unrelated to my talk, but I thought that's interesting too, because you get all these messages how easy some people save human and the other one not, and it's really age related. You know, as younger you are, as more likely you survive. So I have to stay away. I would be dead very 85%. So age is a major factor. And they say in certain areas they have more younger patients. So we have to be a little bit careful if we say there's really now a therapy for Ebola. Okay. And now we come back to the viral load. And what I really liked about that's a complete viral load over time. But if I go here, you see the bold, so these 3 bold lines, they never had any viral load. One of that had no viral load, and this one never had virus. So they were completely protected. If you look here, this, which has a little bit lower antibody, this is this here. And this one really doesn't line up too much, but this here lines up. So the second one has previous short. And if you don't see good antibodies, that's what happens, and you get a lot of viral load, and that was animals which had to be sacrificed. However, now comes, if you look to when we went into the challenge in this animal here and here and here, they are very similar, these antibody levels, right? I mean, this here, what is the difference? And that was what puzzles us. Why do 2 survive and why? It could be CDL responses, but I really believed more on the antibody, and we looked a little bit more on the antibody. And what we really saw here, if you look to the IgG1 to IgG2 in humans and monkeys, IgG1 is Th1 responses, and Th1 responses are necessary to actually have ADCC. So if we look to that over time, after challenge, then you actually see that all the animals surviving here, they go more to Th1, whereas the other one go more to Th2. So we really believe that the antibody response is a major, and it's not significant if you take the replication deficient, but if you take this to this surviving, replication competent, and killed, it's highly significant that Th1 bias is important. And I don't want to say if you don't, certainly the level of antibody is very important too, but I think the quality of the antibody is very important too, and you need more Th1 response. So what else are we doing? I'm almost done. Yeah, I'm still on time, I guess. How do we increase Th1 bias for, say, Ebola GP? So for humans, we really want a killed vaccine. Everything else is, it's just rabies now. Hard to get that approved. So what we think, so we have 2 hypotheses. One is Ebola virus is controlled in large by antibody with antibody-dependent cell-mediated cytotoxicity playing a major role. That's our overall hypothesis. So we think, we don't say neutralizing antibodies are not important, but I think you need both neutralizing and non-neutralizing antibody. We know that rabies virus G has a complete Th1 bias response, not completely, but a very strong. And one theory, it's kind of a desperate one, we think that the high density on the rabies virus G on the surface is important. There's really no good basis in immunology. I still think that structure, recognition of structure is very important in immune responses. At least it makes sense. And in any case, having more of your antigen of choice in there would be a good thing. So we thought, can we improve cooperation of GP and then getting a more Th1 bias and a higher immune response? And what we did for that was codon optimization, something I always thought won't have a dramatic effect, but it has. So we codonized Zaire GP, Ebola GP. This is our vaccine, which I just showed you in this experiment. And this is a codon optimized expressed, and we get about 4-fold more increase. In fact, now in the viral particle, which I'm really happy about, this is rabies G here, and this is Ebola GP. And they equal, it's about 10% now within the particle. And we did one quick experiment, which is also a little bit confusing because we had to use adjuvant. Basically, I got 4 monkeys from the NIH, but they wanted to test new adjuvants, and they said, without adjuvants, we don't test your vaccine. And I said, okay, what can you do for non-human primate? We reduced the amount of antigen, so that's where's a new particle. One used the TLR1/2 agonist, one a TLR3. It's from a company, so I can tell you what it exactly is. They were immunized twice with 100 microgram, and we had 100% protection after challenge. So the question is certainly, can we get it without adjuvants, which would be the preferred way to go, because I learned that adjuvant is really makes your life very difficult with the FDA. So in conclusion, I think we identified the best candidate, at least for rabies virus, with the codon optimized. We think that's a BPL inactivated vaccine is the best we can use. We actually, this started, we in the 3rd year of a grant, and IDT Biological is a contractor in Germany. We already produce Zaire, Sudan, and Marburg in parallel. They were all recovered on approved cell lines, so we can go in production soon. But in the meantime, due to the outbreak, and that's NIH is certainly interested in multiple different vaccines, we got some funding to produce the Zaire strain now, so it's a real Ebola virus of the current outbreak, and that's done end of this month, most likely, master seed we have. We started just a big non-human primate study with 24 animals, where we look for dose dependency and the need of adjuvant. And we also prepare the first clinical trial, also NIH sponsored. Hopefully we know by mid of this year, yeah. You never know. Things take time, and you still have to be careful. I think some approaches may be pushed a little bit too hard before it was too slow. So I hope I could actually couldn't convince this guy, but could convince you that you actually, with one bullet, can kill 2 birds. And I want to thank all the people involved. It's a huge amount of people who are actually involved in such studies. Most of my work is now done at the Integrate Research Facility at Fort Detrick, which is a facility just for researchers to perform experiment with BL4 pathogen. We worked with Heinz Feldman with that last animal study at the Rocky Mountain Lab. We have the production team for the vaccine. This vaccine was now licensed to Excel Bio to drive the commercial part of it, and this group is in my lab who works on the more basic research. Thank you.
Wonderful, thank you. so looking very promising. Do we have any questions from the floor? There should be some mics going around, which I can't see.
Do you foresee the possibility of using this vaccine to deplete the bat reservoir?
It will be very hard. I mean, to immunize bats, I mean, that was a thought with rabies. I mean, you could try, but that would be challenging. I mean, you could bait these bats with certain fruits coated with virus, but I think it certainly could be made, but realistically it would be very expensive probably. And in this area, you probably want to put your resources more into humans, I would think. Because, yeah, would be interesting. I would love to try it, but I think you have a lot of challenging to find some money for that. But it's not impossible to all immunize a bat with rabies. We can do that.
Yeah. In your preclinical studies so far, have you been able to look at route of immunization, like intradermal, to optimize dendritic cell presentation?
No, we didn't. That's a good idea too, because in India they do intradermal immunization with a rabies vaccine just for cost factors. It's a little bit challenging. In India it works well because they have a lot of nurses which can do the TB test. But yeah, certainly that's something for later. Right now we want to try probably just 2 doses of the vaccine for safety in the phase 1.
Thanks.
An aside question from your vaccine research. I was curious, on an early slide, you showed various wild animal species in the U.S. I was curious, does Canada have a rabies surveillance program, and what are the vectors up there?
In Canada, they're very successfully immunized with recombinant adenovirus. You can do that too. And they had some success with that system. There was in the US, there was some try with recombinant vaccinia virus expressing rabies G, but there was some incidents that somebody got unfortunately infected. With rabies, I mean, I have to defend the USDA a little bit. With rabies, the problem is thermostability, so we work on that too. In Europe, it's not as hot, like in the Midwest during the summer, so rabies is not a stable virus, because I think the oral rabies virus is still pretty good. The other thing with oral immunization is the problem, for example, if you immunize dogs, they don't chew a lot. Foxes are good chewer, so if they find a bite, they chew it up. If you have a dog, you know how they eat, they just swallow, so that doesn't help. So there are certainly challenging things. Skunks are incredibly difficult to immunize. Nobody knows why. You can put them 10 to the 10 in the oral cavity and they don't seroconvert. So yes, there's a lot of work to be done.
Question, the differential disease penetrance with Ebola with age, which we see in so many infections with West Nile, for example. One, what do you think is the underlying basis for that, presumably immune response capacity? And the other, the response to these vaccines, are they going to be age-related as well? Will older people respond less well? And
yeah, that's a good question. That's certainly things you have to look into. Why it is, we really don't know. I mean, the normal thing is a very young and a very old but that age is not very old. So I think nobody knows why that is. Perhaps we will learn from the clinical study if that is Africa specific to co-infection, to multiple infection. Do these people have already other infection? I mean, they have quite a lot of viruses there. But that certainly has to be analyzed. And certainly with these vaccines, there needs to be some risk analysis too, because you have people with AIDS and things like that. So that's certainly all concerns.
So I was reading on that most of the antibodies produced for against the glycoprotein or against the mucin region of the glycoprotein. Have you tried building a vaccine without the mucin regions, see if you get more neutralizing antibodies, and what's the difference between?
No, we didn't. I talked to Heinz Feldman. They made a VSV with a mucin deleted GP, and it was horrible. Didn't work whatsoever, so I didn't bother. What we did is actually for our ELISA plates, we have two different GPs. We have one with a mucin domain, and we have one without the mucin domain as a coding antigen. And we really didn't see significant difference in the ELISA titers. So we probably have a different antibody. So in that, we didn't see. But it would be really good to look a little bit more into cell population, what you get. What we try to do is going now in humanized mice to actually look what kind of antibodies we may get there.
So do you have any kind of toxicology studies? Have you looked into the monkey studies with your Ebola vaccine candidates?
Pardon me?
Toxicology studies in monkeys, do you have any chance to look into that with your candidate vaccines?
We have a new study now, but that just started 2 or 3 weeks ago. So I will probably know in 2 months, 2-3 months, we will have the challenge data. Then I know more. But right now, we not even analyzed sera.
Last question, Erica.
The other half the answer for the mucin domain antibodies. A lot of the mucin deletions in the field are badly designed and unfold the protein. So we just don't have a good answer.
Okay.
Great.
Thank
Thank you
you.
so very much.